It's my honor now to introduce Dr. Polly O'Rourke. She's an associate scientist at the Applied Research Laboratory for Intelligence and Security, called ARLIS, which is a university-affiliated research center of the Department of Defense. It's, I understand, physically located at the University of Maryland's campus in College Park, or near College Park. She is currently leading the University of Maryland's performer team in the DARPA Targeted Neuroplasticity Program, in which she is examining the impact of non-invasive peripheral nerve stimulation on language learning. Dr. O'Rourke earned her PhD in linguistics from the University of Arizona, where she specialized in psycholinguistics and cognitive neuroscience. Her talk is entitled Predicting and Influencing Behavior by Way of the Autonomic Nervous System. Thank you for joining us today, Dr. O'Rourke.
Thank you so much for that introduction and for inviting me to speak. This is an exciting opportunity, and I've really enjoyed the talk so far. I am from the Applied Research Lab for Intelligence and Security, a UARC at the University of Maryland. I've been there almost 10 years now, and for the past 10 years, I've been thinking of ways to use the tools and insights of cognitive neuroscience and cognitive neuroscience research to support the Department of Defense and intelligence community. My work has mostly focused on human performance and human performance as it relates to workforce readiness. So looking at selection, looking at training, looking at assessment, and also monitoring throughout a career per se. And what I wanted to talk about today are, some of the insights that I've gathered from trying to use cognitive neuroscience in this way, talk about some of the work I'm doing, and, one path forward in terms of how neuroscience can be used to serve the mission of the Department of Defense and intelligence community. And so when we study cognitive neuroscience, I can't advance my slide. There we go. When we study cognitive neuroscience, we tend to think only of the brain. The brain is where it's at. This is neuroscience. When we're trying to use this research in applied contexts, that becomes rather limiting because there's a lot of wonderful things we can do in the lab in terms of using neural imaging data or psychophysiological data to predict behavior, predict different states. However, that's less viable when you get into the field, when you try to transition it into an operational context or really any time outside of the lab. For example, when we think about aptitude testing like the ASFAB or the DLab, that's never going to involve an fMRI scan. It's very unlikely, just because of a variety of factors, including expense, time, resources, etc. And similarly, when we look at the brain in terms of modifying behavior or improving performance, there are a lot of limitations there, a lot of things that make it difficult in the current state of technology. Some methods are highly invasive or involve pharmacological interventions, which can have unintended consequences. And so thinking only of the brain when we think of cognitive neuroscience and psychophysiology can be somewhat limiting. The picture changes when we consider that the brain is housed in a body and part of this larger interactive system. And so that's what I'm going to be talking about today. And you can see from my little cartoons that I'm drawing out the connection between the brain and the heart. This connection is mediated by the autonomic nervous system. And because, and I'm going to talk more at length on this as I go forward, but we have these two-way reciprocal connections between the heart and the brain. allowing us to get information about psychological states from the body, from recordings that we can easily take from the body. And the question, there's a couple questions if I'm posing this as a potential solution or set of solutions. Can we get similar types of information by making recordings from the body as we can from the brain, however we choose to record from the brain? And two, can we use techniques targeting the body to influence behavior rather than targeting the brain? And so I'm going to be talking about insights from existing research and then some work that I've done to try to make the argument that there is a lot that that by harnessing the autonomic nervous system as a predictor and as an insertion point, we can get a lot of mileage out of it and a lot of more practical, usable solutions in applied contexts. And so here's just a little rundown about these brain-heart circuits that I'm going to be talking about. So here again, we have our cartoon brain, and way at the bottom, much usually overlooked, not considered very seriously, is the brain stem. The brain stem is an extremely critical part of the central nervous system, not only a relay between the body and the brain, but there's a lot of important regulatory functions, and there's a lot of aspects of higher cognition that are underpinned by the circuits involved in the brain stem. From the brain stem project 12 specific sets of nerves called cranial nerves. These nerves, their main function, one of their functions is to innervate the head and neck, providing both motor and sensory innervation. And some of them, particularly the vagus, cranial nerve number 10, has a lot of regulatory functions in terms of controlling the motor and sensory information, or innervating, providing motor and sensory innervation of the viscera, heart and gut. So now we're getting back to something that was mentioned earlier about this whole system being composed of the brain, the heart, and the gut. That's kind of the crux of the critical nature of the vagus nerve. And so here we have a close-up of the brain stem showing the roots of the vagus. As I mentioned before, the vagus is providing sensory information for different parts of the head and neck, specifically the ear, but other parts as well. Visceral sensory information and visceral motor for the heart and the digestive tract. It's also controlling the balance between the sympathetic and parasympathetic nervous system. And so when I'm talking today about the autonomic nervous system, the whole system is not just these vagal circuits, but these vagal circuits are a big part of it. And so I'm going to be focusing on that today. When we talk about the parasympathetic versus sympathetic nervous system, we're talking about the regular way in which we shift back and forth between a comfortable default state, which we'll call rest and digest, which indicates parasympathetic control. This is when the individual is calm and not feeling threatened and able to go about their business. And the other state, which we kind of term fight or flight, is a moment of stress when the individual feels threatened, when they've experienced some kind of stressor that makes them react. And in our normal lives, we're always kind of flip-flopping back between some degree of these states. We hopefully every day don't go into full flight or fight where we feel our lives are in danger. But as we experience small threats, whatever they may be, our autonomic nervous system is helping keep us, helping us respond appropriately to threats and return to the default state in a flexible way. This is mediated by the vagus. And one of the major circuits involved in this function is this brain-heart circuit. Obviously, when you experience a threat, that causes a change in heart rate in order to provide the appropriate, metabolic response. And that change in heart rate, there's these two-way connections. So if your heart is acting stressed, that feeds into your stress response at higher levels in your brainstem and brain. If you feel psychological stress, that's going to further change your heart rate. So this is a two-way system, and this is kind of, one of the bases of the connection between stress and anxiety and heart disease, where heart function is affected by psychological factors and vice versa. And the way your heart rate fluctuates in response to stressors is indicative of your general ability to regulate stress and regulate your emotional responses. So what the autonomic nervous system is helping us to do is to provide this kind of regulation. Happily, when we look at data recorded from the heart, when we record heart rate, we can capture that interaction by looking at how the intervals between heartbeats vary. That's called heart rate variability, and that can tell us, how strong our parasympathetic control is, or if we're having a more sympathetic fight or flight response. There are a variety of dependent variables that can be extracted from heart rate variability. And through looking at this data, through the ways we can analyze this data, we can have a measure of parasympathetic control. And that is a proxy for a stress resilience, among other things. So this is an interesting physiological measure. And then the question is, okay, so that's great. We have this measure. It tells us something about the heart functioning, maybe something about the stress response. Are there other things, other information that we can get from these vagally mediated circuits? What else is the vagus doing? Why am I trying to sell it as a kind of an insertion point for modifying behavior or a psychophysiological metric? So when we look at the vagus, we see that its peripheral branches project into the brainstem, hitting specific brainstem nuclei, critically the locus ceruleus, which is the brain's only source of norepinephrine, but others, the pedunculopontine nuclei, which transmit acetylcholine, and the raphe nucleus for serotonin. Collectively, these circuits underpin the top-down control of attention, acquisition of declarative knowledge, also shifting between scanning and focus states of attention, and mood and pain modulation. So the vagus is critically, in addition to these kind of lower level, arguably lower level regulation circuits, the vagus has, the functioning of the vagus is going to also influence cognition. And then we would expect that to some degree, cognitive state may be represented in the heart rate data because it is this two-way circuit. So getting back to my earlier question, this is all great, but how much can heart rate variability really tell us? So the answer is quite a bit. There's quite a lot. There have been a number of studies looking at, contrasting heart rate in individuals, looking at it as an individual difference factor or as a between subjects factor. And what has emerged from this body of research is that there are quite a lot of distinctions we can make based on heart rate variability, and specifically the metrics that indicate degree of parasympathetic control or stress resilience. So we can predict by recording someone's heart rate, we can look at their trait stress resilience, how generally we would expect them to react to stress. And also we can, when they're doing a stressful task, we can determine how stressed they are by looking at that data. Also, we see differences in heart rate in individuals, heart rate variability in individuals who have depression, in individuals who are suicidal, and in individuals who have substance abuse issues. So, it is a predictor that can predict some outcomes that are very relevant in, most workforce contexts, but specifically in the military, these are issues that decision makers want to know about in their workforce. Importantly, for the cognitive neuroscience folks, and again, this is, why I'm discussing this as a psychophysiological metric, there's a lot about cognitive ability and cognitive performance that we can learn from heart rate data. So there have been a variety of studies showing that heart rate variability can predict working memory capacity, it can predict attention control ability, performance on creative problem-solving tasks, performance on language learning tasks. And this is just a subset. There's an emerging body of research showing this. So, while heart rate variability obviously does not give you the types of resolution that, neuroimaging data will give you. For example, obviously, you can't really say very much about the neural bases of what's happening when a person is performing these working memory tasks. We're not getting actual data for this. There's not an fMRI scan that we can point to and say, oh, it's prefrontal. And also, we don't have the temporal resolution of a method like EEG, where we can, look at processes that occur very, very quickly and gain insights into processing. Nevertheless, this is an indicator that's easy to collect, inexpensive, and can provide you some key insights that could be fed into decision-making systems or given to decision makers for manpower decision or selection, etc. Now, having said all that, the underlying constructs of the relationships between heart rate variability and the various outcome measures are not entirely clear. And it's possible that it's a reflection of the interplay between stress resilience and cognitive ability. That's not exactly clear. Nevertheless, you can extract pretty interesting information from this physiological metric about an individual. Right now, I think from what I see, kind of perusing the calls for funding and the research interests of places like DARPA and IARPA and others, there seems to be a lot of interest in making sense of this rich, continuous sensor data that we're now increasingly collecting on warfighters. And so I think, one future direction is developing the machine learning algorithms and developing perhaps the artificial intelligence type analytics in order to extract the maximum utility out of this data to support operations or whatever the context. Okay, and now taking a shift towards influencing behavior. There's some old ways that we think about influencing behavior and some newer ways that I'm going to talk about, or ways that are new to me. So take a deep breath. You know how when someone is upset and they start to panic and you say, take a deep breath, and that often will help. This circuit is really what you're doing when you take a deep breath, that you're deliberately slowing down your heart rate. Like taking a deep breath, a slow deep breath will slow down your heart rate and increase your heart rate variability. And that, information gets to your brain and reduces your stress response. So you're doing kind of a deliberate technique for impacting that circuit. From a more systematic perspective, there are other ways to do that. This is cardiovascular training. Will increase your heart rate variability and therefore, increase the health and strength of that vagal circuit for emotional regulation. We all know how easy it is to increase our physical fitness, right? That's something we all do effortlessly. That's a technique that's going to require some work. Another way, I'm going to skip ahead to meditation. Meditation involves controlling the breath and also trying to deliberately control arousal, kind of psychological arousal. So slow controlling breaths, therefore increasing heart rate variability and trying to create this state of low arousal. Maybe a little bit easier than going for a brisk run, but also requires time and effort. Biofeedback training is an interesting technique where individuals deliberately breathe at the frequency that maximizes their heart rate variability, and that can be determined with sensors. And so there's no cognitive component. There's no need to get into a specific mental state. It's simply controlling the breath to alter the heart rate. Research indicates that all of these techniques work and that there are, the predicted cognitive and health benefits to them. One drawback to them, as to many types of interventions, behavioral and otherwise, is that all of them require some effort on the part of the user. And in many contexts, that's fine. People have time and energy to devote to these things, but a lot of times the more effort that's required, the less likely that the user will end up complying with a given regimen. So another way to influence the vagus and the corresponding autonomic circuit is peripheral nerve stimulation. So actually targeting the vagus with electrical, principally electrical, but there are other techniques for stimulating. This is a technique that's been used quite a bit in clinical research, principally for epilepsy, but other neurological conditions as well. In patient research, it typically involves a surgically implanted stimulator that's wrapped around the cervical branch of the vagus to provide stimulation controlled by a medical professional. When you talk about performance enhancement in healthy adults, having a surgically implanted device is less desirable. Obviously, it comes with some risks, and that's not something most people will do unless it's to alleviate an acute medical condition. The vagus innervates parts of the outer ear, including the outer ear canal. Peripheral branches of the vagus stimulate the outer ear canal. So it can be stimulated by placing an electrode on various different parts of the ear, including electrodes that are housed in common-looking earbuds, can be inserted into the ear and stimulate these peripheral branches that propagate to the brainstem in ways I've discussed before. This is kind of a new body of research, particularly with the auricular stimulation. And it's being explored not only to replicate the clinical findings from the implanted devices, but also looking at performance enhancement. And here's just an example. This is one auricular vagus nerve stimulation device that's commercially available. You could go online and buy one tomorrow. And you can see the normal looking earbuds and the very small device, basically a wearable device. So this is something that, if it's effective, could be broadly implemented. Doesn't require any effort on the part of the user other than putting the electrode in their ear and turning it on. So it's pretty exciting in that if it's effective, it could easily be implemented to broad populations. And that brings us to the work that I'm doing on DARPA's Targeted Neuroplasticity Training Program, where we're looking at auricular vagus nerve stimulation as a technique for enhancing language learning. This is part of the larger TNT program where there are multiple teams all over the country or across the country looking at vagus nerve stimulation, transcutaneous vagus nerve stimulation to enhance cognition. And I'm going to just do a quick overview. This work has been going on for a little over 3 years now, so we're in our final year of the program. And we've gotten some exciting results. So I'm going to just present a small subset and tell you first, just kind of set up our methods very, very briefly. I can give you an 150-page tech report if you want the full full information. But we're delivering stimulation through the ear before a vocabulary learning task, during a learning task, or during recall testing. So I'm going to talk about 3 different administrations of the stimulation. The control group receives no stimulation, and we're delivering the stimulation below perceptual threshold. So we calibrate it beforehand to determine for each individual, for each session, at what level can they actually feel the stimulation, then we stimulate below that. We did that, number one, to help maintain the double-blind design, and number two, because we wanted to develop a technique that would not inconvenience the user at all. Just they wouldn't even know they were getting it, and therefore it wouldn't disrupt whatever activity they were involved in while they were being stimulated. And our participants were the usual suspects, students and community members, students from the University of Maryland and some members of the surrounding community between the ages of 18 and 40. So here are the results from one experiment where we looked at the TVNS delivered before the learning task. So participants received 10 minutes of stimulation, and then they did a word learning task. And what we see here is from the first training session to the second, participants in the stimulation group got faster. Their reaction times in this translation recognition task were reduced relative to sham. And I mean, that's interesting, but there's a lot of ways we could analyze that. We could interpret that. Is it just some kind of like psychomotor thing, or are they actually somehow creating more developed word representations? For this study, we collected EEG data, and when we looked at that, we saw that the stimulation group, again, that group that received stimulation before the word learning task, they showed a potential called the N400, which is associated with abstract word representations in the brain. And so the stimulation group had what looked to be a well-developed N400, while the sham group had none. So in addition to the speed of their response, it seems like stimulation was leading to accelerated formation of mental representations of second language words. In another experiment, as I mentioned before, we looked at stimulating the participants during the word learning task. So in the word learning task, they're exposed to a number of word pairs that they're told to remember. And what we saw when they received stimulation during the task, during the training itself, they had greater accuracy than controls. And so this figure here is showing us their accuracy in a translation recognition task where they see two words and are asked to determine if they're translation equivalents. And so when they are translation equivalents, the sham and the treatment group look about the same, but when they are not, the treatment group is much better able to make that discrimination and say, no, these are not a match. The sham group does more poorly. Another experiment we did suggested that the effect of this type of stimulation during training might be maximal for words that have high frequency English translation equivalents. So learning the Indonesian word for dog might be easier, it might be more enhanced by TVNS than learning the Indonesian word for anvil. So, basically what we are seeing that we can enhance word learning, second language word learning, and that there may be some distinctions related to the lexical features of those words. So very exciting. We induced a behavioral change with our intervention, and it was in the direction we had hoped. So big success. When we looked at the data from the participants who received stimulation during recall testing, so in a recall test, they've completed the training phase, and then they see a foreign language word, and they have to type the English word. In the recall testing, we saw that participants who received stimulation during the recall testing did more poorly, had more shallow learning curves than the sham group who received no stimulation. So that's pretty interesting, obviously a little disappointing, but very informative. And this gets back to what Dr. Flynn said right in the beginning about separating the fact from the hype. Because when we went into this endeavor, there's a lot of optimism about what TVNS can do. And based on the neural circuitry and on our hypotheses and those of others, we think it could do almost anything. However, what we're seeing is not only are there, of course, going to be limitations in the magnitudes of the effects and the specificity of the effects, but also it can actually harm performance when administered in certain time frames. So that was very interesting. We also looked at mood and anxiety, and we included these tasks because, again, based on what we know about the connections between, this parasympathetic and sympathetic tension mediated by the vagus and stress and negative mood, we wanted to look at how experiencing the stimulation changed these levels. And so prior to every TVNS session, so right before and right after, we did mood and anxiety scales for our participants for each session. And what we saw was that while the sham groups showed increases in negative mood and increases in cognitive anxiety, the groups, this is in particular, this is the data from the participants who received the stimulation before the learning tasks. Those participants either showed reductions in the case of cognitive anxiety or stayed about the same in terms of negative mood. So we are seeing an impact on mood, which is very interesting and also encouraging because it fits into a larger story and also the kind of larger emerging literature that this intervention can have positive effects on mood. And just a side note, that it was safe and comfortable. We had elaborate questionnaires looking at comfort and kind of user experience. And the only thing that individuals who received stimulation reported at a higher rate than sham was itchiness. So their ears were a little bit itchy, and that was it. So we were really encouraged by this because ultimately, if we want to implement anything, on a broad scale, it has to be, minimally, obviously no greater than minimal risk and ideally comfortable. So we're very pleased with these results, and we're continuing to do this research with the goal of, validating and having good recommendations for use for the safe, inexpensive, basically wearable, and intervention that requires almost no effort on the part of the user, and they can receive benefits. Okay, and finally, and again, we're getting back to what Dr. Howard had mentioned about the brain, the heart, and the gut. I've been focusing on the heart, but the vagus also is involved in these autonomic regulatory processes in the gut viscera. And that is another interesting insertion point that hasn't been exploited too much, but I think, again, this is something that's kind of emerging as an area of interest. And when I talk about the gut, I'm mostly talking about the microbiome. The microbiome, as I'm sure many of you know, is this complex ecosystem of trillions of microorganisms that support digestive function. And their role goes beyond that, but putting it simply. And digestion and lots of features of the digestive system are regulated by these two-way vaguely mediated connections. And the gut microbiome, I mean, I've read some scientists have construed it as like the critical underpinning of stress resilience. I'm not sure if it wins the prize. I don't know if anyone does, but it's very sensitive to stress. In that, when an individual is stressed, it causes changes in the lining of the gut, which can cause the microbiota to pass through and then have a negative influence, such that a stress response is induced by that, which in turn stresses out the body more. So again, these two-way connections. And there's a lot of research, there's an emerging body of research looking at the microbiome and stress resilience, and it's possible that there will be some connections between that and cognitive performance as well. It presents an interesting insertion point, and there are ways you can influence the microbiome, using dietary supplements and things like that. So that might be another emerging area. And again, there's a big data analytic challenge because of the magnitude of the data that you would get when you look at the microbiome, and trying to gain specific insights from it will require a lot of sophisticated modeling capability. So that's one more, just kind of as a side note, a way to influence the system and autonomic function. So general conclusions, there is a wealth of information we can gain from indices like heart rate variability and possibly microbiome measures and other things like galvanic skin response. I've kind of limited my discussion to heart rate variability, but there are other physiological metrics we could also exploit that can give us a lot of information about an individual's cognitive abilities and function. And I know that there's an increasing use of all kinds of sensors in warfighters and others, and a need to make sense of this information. And so there's a big opportunity right now to give real purpose to collecting that information. Obviously, it would be very contextual, whether, depending on what you need to know about an individual in an operational context to optimize their performance or reduce risk. But a lot of opportunity there for this type of information to support mission. And autonomic function can be targeted to lead to enhancements in cognitive performance and health and mental health generally. And harnessing, these two aspects can support the Department of Defense in a variety of ways. And I've talked about selection, training, manpower decisions, et cetera. And could be a real, I think it's a moment where we have an opportunity to conduct some really exciting research to bring this into the field and out of the lab. And that's it. Thank you very much.